Hey, my name is Zaylee Kennedy, and today we're going to be talking about lysogenic conversion and how it turns ordinary bacteria into pathogens. Two key terms in this is going to be your prophage and your provirus. A prophage is the genome of a bacteriophage, a virus that infects bacteria. When it's integrated into the DNA of a bacterial cell, it lies dormant but can later become active. A provirus, on the other hand, is the viral DNA of an animal virus inserted into the genome of a erotic host cell. The key difference is prophages infect bacteria while proviruses infect animal cells. Next, we're going to be talking about latency and lysogeny. So, how is latency in animal viruses different from lysogeny in phages? In latency, the viral genome can remain active in a host cell for months or even years without integrating into the host genome. Think herpes viruses hiding in the nerve cells. But in lysogeny, the phage genome must go into the bacterial chromosome, becoming a prophage that replicates along the host cell's DNA. So next, let's look at three major differences. Destruction of host cells. Lytic cell, the phage takes over the host's machinery, makes new viruses, and lyses kill the cell. Lysogenic cell. The phage's genome integrates and the cell stays alive, continuing to divide. Another one is viral replication timing. Lactic, immediate replication in assembly. Lysogenic, viral genes are dormant and do not replicate immediately. They replicate along the host of the DNA. Next is going to be environmental triggering. Lysogenic viruses can switch to the lactic cycle in response to stress, like the UV light or chemicals, while lactic viruses act quickly and destructively right after the infection. So, lysogenic conversations happen when a prophage brings new genes into the bacterial genome, especially genes that make the bacteria more dangerous to the human. In other words, viruses give bacteria new weapons. These germs might encode toxins, enzymes, or surface proteins that make the bacteria more infectious, harder to kill, or resistant to host defenses. So next, let's talk about examples of lysogenic conversions. So two classic examples is Corynebacterium diphtheriae. Alone, this bacteria is harmless, but when it carries a prophage encoding the diphtheria toxins, it causes diphtherium, a serious respiratory disease. Another example would be Vibrio chloride. This bacteria only becomes pathogenic when infected by a phage that carries the gene for chloride toxins, a key factor in deadly diarrheal illness, chloria. So therefore, lysogenic conversion is a powerful tool, turning friendly or neutral microbes into serious health threats. Understanding lysogenic conversion is getting a deeper look into how viruses and bacteria evolve together, and why controlling viral infections can be key to preventing bacterial diseases.